My name is François Thomas and I'm a scientist at the Woods Hole Oceanographic Institution. I'm working as a postdoc in Stefan Sievert's lab and I'm studying microorganisms in a salt marsh. And most of my work is done here in the laboratory, but we need to go also in the field to sample. And today I would like to take you to a local salt marsh here in Woods Hole. So microbes are organisms that are too small to see with the eye and you need a microscope to see them. And they're actually really important for the ecosystem. So the project I'm working on right now is about salt marshes and what microbes are doing in salt marshes. Salt marshes are like a buffer between the land and the ocean. So basically whatever happens here in the salt marsh controls what will go from the land to the sea. So with the increasing use of the coastal environment, you have much more input of nutrient in the salt marsh because of agricultural use or wastewater treatment. So the microbes that are in the sediment here are trying to keep up with the more nutrient loading, and we're trying to understand how they do that, and depending on the conditions, will they be able to restore the environment to its initial state or not, and what will happen to the ecosystem if they don't. Here we are back in the lab at Woods Hole Oceanographic Institution, and we're going to take the core out of the tube. So I'm cutting the core in half to get access to the interior of the core, and here you can see how dense the root system is. So here I found a root, I'm cutting it, and I'm going to put it into this tube, which contains a solution that will preserve the microbes. So to try to maximize our success of growing different microbes, we try to use different conditions. So even if we try very hard to grow all the microbes, we can't have access to all of them because some of them won't grow in the lab, because we don't know what food they need, for example. So to try to still identify all the microbes that are there, we use other techniques that we call molecular techniques. To do that, we have to extract the DNA, which is the genetic information of the microbes, from the sediment. Using this DNA that we just extracted, we're going to read a particular region of the genetic material, which will allow us to identify all the microbes that are there. So we have some results from the work I've been doing in the salt marsh so far, and one of the most interesting results is that we can actually see differences in the microbes that are growing in the sediment compared to the ones that are growing on the roots. So in this graph, each dot represents a sample that I took in the salt marsh. The blue dots are root samples, and the red dots are sediment samples. The closer the dots are, the more similar the microbial community is. And in this 3D graph, you can see that the root samples in blue are closer to each other than they are to the sediment, showing that there is really a difference between the microbial community living on the roots compared to the one living in the sediment.
Where are you originally from?
So I'm originally from France, where I got my PhD in microbiology and biochemistry, and I've been here for a year and a half, and I enjoy it. Is that too much?
No, leave it. You gotta leave it. Now you have to leave it.
No, that's not.
Okay.